right, in this video we're going to look at DNA and DNA replication. This is video 6 from chapter 4, again covering for our PowerPoints at Or Georgetown Tech. The slides basically 34 through 43 in this. Now, DNA, deoxyribonucleic acid. DNA is our genetic material. It's where a lot of DNA is going to be made up of these areas that we'll look at called genes that are the information to make different proteins. Now, as it says, DNA is a molecule that stores information mainly on how to synthesize proteins. We've got some of the proteins you should know, or some of the things about proteins you should know is proteins make up enzymes and antibodies for sure. They make up some blood proteins, albumin stuff, that's a type of a protein. But the main ones, again, if I'm going to list things like proteins, carbohydrates, nucleic acids, lipids, things I want you to know about proteins, what proteins make, two main ones, enzymes and antibodies. But anyway, the DNA is where we store the information to make a protein, and we call that section of the DNA a gene. Now, the entire genome is the entire set of genetic information in the cell. So we have this recipe book called DNA. The entire list of recipes is called the genome, and each individual recipe would be called the gene. The exome is the small portion of the genome that codes for proteins. Now, in every cell in our body, we have the same molecule of DNA, but different cells will produce different enzymes. For instance, certain things in the liver are going to do, they're going to produce different enzymes and different proteins than, say, the eye, certain cells of the eye, which are going to produce certain proteins. Now, each one, because each cell has different genes that are working, we call that gene expression. Now, most of us come into the class with the prereqs understanding that DNA is what we call a double helix. So it's a two-stranded twisted molecule. So we have two chains of nucleotides. Now, DNA resembles a ladder twisted in the spiral, a double helix we call it. And there are bases from the two complementary strands that are linked together by hydrogen bonds. We'll look at the names of these in just a second, but remember that C always binds to G and A always binds to T, or vice versa. And these are called nucleotides. Or these are called nitrogenous bases. But when we look at nucleotides, remember nucleotides are the building blocks of nucleic acids. And each nucleotide is going to have a sugar. In DNA's case, it's called deoxyribonucleic, so that's where we get deoxyribonucleic acid. It's going to have a phosphate group, and then it's going to have this nitrogenous base, and this is where we get the A's and the T's and the C's and the G's. Adenine, cytosine, guanine, and thymine. Now, hopefully, this adenine kind of strikes a little bit of a memory bell because we talked about adenine triphosphate, adenosine triphosphate. Anyway, this is how we get in the ATP is part of the nucleic acid group, but that's for another story, not meaning to get sidetracked there. Adenine, cytosine, guanine, and thymine are the four nitrogenous bases. And remember that adenine always binds with thymine in DNA, and cytosine always binds with guanine. This is a nucleotide structure. I should have taken this slide out. It's just simply talking about the phosphate, the sugar, and the base. Now, in the one strand, these are little strands of nucleotides. And what happens is the nucleotides from one, or the nitrogenous base from one nucleotide to the base of the other form a hydrogen bond. So remember, anytime you see these little dotted lines, that represents a hydrogen bond. And again, you can see anywhere there's an A, there's going to be a T on the other side, or vice versa. Anywhere there's a C, there's going to be a G, or vice versa. And so this is how they bind together. Remember, those hydrogen bonds are just strong enough to give it some structure, but not too strong that it can't be pulled apart, opened up, read, and then closed back together. And we'll see that later on in this chapter. Now, the two nucleotide chains form a double helix, and they run in opposite directions. It's referred to as anti-parallel. But again, remember, A and T always bind together, and C and G always bind together. If you get farther along, and then now this is not something I'm going to test you on, but if you get farther along, A and G are purines, and C and T are pyrimidines. Now, a purine will not bind with another purine, it binds with a pyrimidine. And again, certain ones bind with certain ones, and they don't mix. Now, as DNA, again, is this super long molecule, it wraps itself and coils itself around certain types of proteins, and it ends up forming the double helix that we're familiar with. Again, this kind of shows how it's all coiled around these little balls, and it kind of condenses up into what we commonly refer to as a chromosome. Now, in the cell, we've already looked at this back in chapter 3 when we looked at mitosis, that when the cell is created and it starts interphase, that is, it's just, I've had a cell just go through mitosis. I've got two daughter cells, so we take one of the daughter cells and look at it as it starts interphase. We have that first little section that we call G1, and the cell's just kind of looking at all the structures it has to see if it needs to make anything else. But then it went into that S phase, and we talked about how at that phase, the DNA is going to make a copy of itself, and we call that DNA replication. Now, DNA replication, again, occurs during interphase, and the DNA will actually start splitting at those hydrogen bonds. It'll unwind and separate, and the nucleotides from each side will start being put together, and they end up making two identical strands of DNA. And this is kind of the picture showing it as it's going through. And when you really understand what's going on, it is so amazing. Again, DNA is almost a 6-foot long molecule just on its own, without having, on a normal scale. Like I said, to imagine if I was to take a basketball, if I was taking a cell to enlarge the cell to where the nucleus was the size of a basketball, the DNA inside of it would stretch almost from Myrtle Beach to Columbia, a couple hundred miles, over 100 miles, and it'll be, it's all in that basketball wound up, and it has the ability to actually split, make copies, and reorganize, which is just astounding. But that's DNA replication, and that's a lot different than what we're going to look at in the next little section. We're talking about how the DNA is read in order to make protein. All right. I hope that makes sense, and have a wonderful day.